Okay, so in today's class, I will be starting with transcription. So before we start with transcription, let me present my screen. Okay, so the first thing is what I need to explain before is the central dogma. So the process by which DNA gets converted to RNA, it is known as transcription. So, what is the central dogma? So, the central dogma is that DNA gets uh, converted to RNA through the process of transcription. So, that is the transcription. That's the process of transcription. So, what you have is the DNA over here. This is the DNA. DNA. From DNA, there is a transcription which takes place, and then a translation, mRNA is formed, and then a translation process takes place, and a protein forms. And the protein is also modified, transcript is also modified. So, this mRNA is also known as transcript. So, this is the central dogma of eukaryotic model. So, today we will be understanding the process of transcription, how DNA becomes mRNA. So the process of transcription. So the key question is, what is the central dogma which I explained right now? What are the four main types of RNA molecules in the cell, and how is an RNA chain synthesized? And how is transcription initiated, elongated and terminated in bacteria? So, as the definition goes, transcription is the synthesis of a single stranded RNA copy of a segment of DNA. So, this is the process of transcription, a DNA giving rise to a messenger RNA. So, the four types of messenger RNA which is present in the cell. First is the messenger RNA. The function of a messenger RNA, as you have read before, is to encode the amino acid sequence of a polypeptide or a protein. So, the whatever is present on the messenger RNA, depending on the sequence of messenger RNA, you have that same sequence of amino acid coming out in that polypeptide. So, mRNA are the transcripts of protein coding genes. So, the translation of an mRNA on the with the ribosome, it produces a polypeptide. The next RNA which is present is the ribosomal RNA. So, the ribosomal RNA is with ribosomal proteins. So, it's in the ribosome. Therefore, it's ribosomal RNA. It's present in the ribosome. Along with ribosomal proteins, it makes up the ribosomes. And the ribosomes are the structures on which this mRNA is translated. So, the ribosome is made up of ribosomal proteins, as well as it is made up of the ribosomal proteins, as well as it is made up of a ribosomal RNA. And this ribosome, it translates the messenger RNA. The next is the tRNA or transfer RNA. The tRNA or transfer RNA brings amino acids to ribosomes during translation. So, these things you would like to read more about these transfer RNAs when you are studying translation. Apart from that, you have small nuclear RNAs present with proteins. They form complexes that are used in eukaryotic RNA processing to form functional messenger RNAs. They are used in translation. So, let us go to the transcript process. So on the gene there are sequences known as gene regulatory elements, and the enzyme which makes messenger RNA from DNA, or makes RNA from DNA, is known as RNA polymerase. It catalyzes the process of transcription. So let us go to figure 1. So this is the transcription process which is taking place. The DNA double helix is denatured by the RNA polymerase in prokaryotes and by other proteins in the eukaryotes. So you can see that this is the DNA double strand. So it is denatured. In this region it is denatured, in this region. So it is denatured in that region. And what happens? So this is the 5 prime. I am drawing the 5 prime to 3 prime end. I am just overwriting this. So I have just overwritten it over here. So that is one strand. This is the other strand. This is the other strand, which is red. So you see on the blue strand you have the 5 prime end over here, and on the blue strand you have the 3 prime end over here, and on the red strand you have the 5 prime end here and the 3 prime end over here, since it is arranged in a double helix in a coil. And now when this so the strand which is then RNA polymerase then catalyzes the synthesis of a single stranded RNA chain beginning at the transcription start site. So there is also a transcription start site. So this place is the transcription, start of transcription takes place here. So on the strand, this strand, the 5 prime 3 prime strand is not the strand which is being transcribed. It is the 3 prime 5 prime strand which is being transcribed, and that is known as the template strand. The antisense strand is known as the template strand because it is on this strand that your RNA transcription starts to take place. So that strand is in the 3 prime to 5 prime direction. Therefore, opposite to like DNA replication, opposite to it, the transcription direction is only one strand is transcribed. In DNA replication, both the strands are replicated in both directions, but in transcription only one strand is replicated, and that is this particular strand. So this molecule which you see here is the RNA which is being transcribed. It is same, addition of nucleotides is same. In DNA replication you had D NTPs coming and sitting. You had D NTPs coming and sitting. You had D NTPs coming and sitting in DNA replication. Over here you have NTPs coming and sitting. You have nucleotide triphosphates coming and sitting on this place. So this is how this is the basic difference which you can find over here. Now, who are the NTPs? ATP, GTP, you don't have a TTP, you have a UTP in RNA, and you have a TTP. These are the nucleotides which come and sit. When this you had DNA polymerase, over here you have the enzyme, this orange colored shaded enzyme known as the RNA polymerase. They are the RNA polymerases. And this particular, let me change the color into something different. Let me make it into, say, light blue. This color has been taken now. Let's make it this color. So this region which you see here, this region, this is a region which forms the RNA DNA hybrid. So that forms this region. It forms this region, you see, an RNA DNA hybrid. So the chain keeps on extending in this direction till a final 5 prime to 3 prime mRNA is formed. So remember the thing that double-stranded DNA in the region of the gene has got two types of strand. The first is strand is known as the template strand, and the opposite to the template strand is the non-template strand. The non-template strand is the strand which is not transcribed. So it is the template strand, which is also known as the antisense strand. It is in the 3 prime to 5 prime direction. It is in the 3 prime to 5 prime direction, and on this there is an unwinding takes place by the RNA polymerase, and in a region of known as on the DNA known as the transcription start site, there you can find the transcription initiates, and the direction of transcription is in the 5 prime to 3 prime direction, and nucleotides are added like NTPs like ATP, GTP, UTP and CTP. They are added. In replication you had DNTPs being added. Here you have NTPs being added. Ribose sugar, deoxyribose sugar. So since this is an RNA which is being formed. So this is the basic process of transcription. So if this is the, say this is the template strand, this is the template strand, this is known as the template strand, and this is the DNA. It is in the 3 prime to 5 prime direction, as you can see. What will be the opposite? The mRNA which is going to be made is this. In the mRNA is going to be made in the 5 prime to 3 prime direction, as you can see over here. So opposite to A, instead of T there will be U. Then opposite to T there will be A. Again opposite to A, instead of T there will be U, because there is no T in on RNA. Then opposite to C there is G. So it will just be the complementary. It will be complementary here. So complementary to the antisense strand or the template strand. Now on this opposite side, on the double stranded end, the other strand is known as the non-template strand or coding strand. It is known as the coding strand because this mRNA sequence over here, that is the same as that of the coding strand or the other strand which is in the 5 prime to 3 prime direction. only instead of T you will find U. Therefore, that is known as the coding strand, because the mRNA sequence which comes out is same as that of that particular strand of DNA, the coding strand of DNA. Only difference being that instead of T you will find U. That is the only difference. Otherwise, the sequence is the same. And the antisense strand, or the 3' 5' strand, the template strand, it is essentially on which the mRNA is going to be synthesized. Sense strand means the word sense comes because that is the coding strand. It makes sense. And the antisense is just opposite of sense, ulto sense. And therefore, it is known as antisense. So let us look at what are the chemical reaction which is involved in the RNA polymerase catalyzed synthesis of RNA on a DNA template strand. So this is what we are going to study right now. So now let us look. This is the DNA template strand or the antisense strand. Direction, देखो, sugars are in the ulta direction, it is in the opposite direction. So it is in the 3'. Let's go below, and 3' to 5' direction. So you have the base thymidine here. These are the phosphates. T, G. This is, we are talking about D. A. This is the DNA. C, T. In DNA you have T. So the RNA polymerase, like DNA polymerase, it will also form a phosphodiester bond. Same thing like in DNA replication. The same, except you are making an mRNA. It will not be a double strand. So you find the opposite to T, you have an A. Remember, in DNA, the 2' end you had only H over here. But here you have OH, since it's a ribose sugar on RNA. That is one difference. Okay, remember this part. And otherwise the structure basically is the same. You have this 3' phosphodiester bond, and you have the G coming here. Only the note that this is a ribose sugar, it's GTP, and over here you have DNTPs on the DNA. The phosphodiester bond is coming here. Instead of T comes U. Also you can see it's a double bond formed. So this region which you find over here, this is what is a short region of RNA DNA hybrid. It's not a double stranded DNA or a double stranded RNA. This is a RNA is to DNA hybrid. Also notice that in this chemical reaction, the direction of the growing strand is in the 5' to 3' direction. So 5', 3' direction of growth. So opposite to G over here you will find C coming. Opposite to T you will find A coming. So like this, as you can see, because of RNA polymerase here, this UTP started coming over here. This UTP came and it got integrated here. And what happened was that these two phosphates got removed. Only one phosphate remained. Only this phosphate remained, alpha phosphate. This is on the alpha phosphate, beta phosphate, gamma phosphate. So these two phosphates, they got removed. So these two phosphates got removed, and you have only one phosphate remaining, and so chain growth plus these two phosphates are removed, and this removal of this phosphate, it like in DNA polymerase action, this gives energy to the reaction. So let us see the transcription in bacteria first. So what are the things present in, let us we are considering that of E. coli. So what you have, let us talk about initiation of transcription at promoters. Promoter is basically a DNA sequence. I will just come to it. But what are the elements which are present? You have a promoter sequence, and you have a terminator sequence, and in between you will have the coding region. Now we are now talking about the DNA only. Now the RNA has not come. The region which is going to be transcribed on the DNA, we are talking about them only. So let us have a look at the figure 3. Let me increase the size so you can see it well on your mobile. So this is the DNA. One is in the 5' to 3' direction, 5' to 3', that is the non-template strand, and the other in the 3' to 5' direction is in the template strand. So the DNA is going on the chromosome or on the DNA. You have before, this is the RNA coding sequence. This is the sequence which will be made into RNA. This light green region, it's going to be made into RNA. This region is not going to be made into RNA. This region is not going to be made in RNA, but they are very important for transcription. So before this region, you have a promoter. This promoter is the region on which your RNA polymerase looks for promoter region. RNA polymerase binds. Otherwise, RNA polymerase will not know where to start transcription. Once it finds the promoter sequence on the DNA, okay, this is where we start our work. It will start its work. It can be on the coding, non-coding, both of them. They can be interested in it. So this is on the 5' to 3' is the non-template, as I mentioned, and the 3' to 5' is a template, and the 3' to 5' strand is going to be used as a template to make your RNA. Then the region where transcription starts, that is known as +1. So before +1 there is no 0. Directly it is -1. Inside the promoter it is -1. So there is no 0. Remember, there is no 0. +1, +2, 3, 4, 5, 6, 7, 8. And before +1 it is -1, -2, -3, -4, -5. It goes in this direction. So the +1 or the first nucleotide which is going to be the region where the first nucleotide RNA nucleotide is going to be added is the transcription initiation site. So the transcription will start from this site, that is the +1. Before that it is the promoter -1, that is not going to be made into RNA, but +1, +2, plus how many, whatever it is, that is going to be made into RNA. So the region before +1 is known as upstream of the region, and this is the region, this is where the transcription stops. So only this part is going to be transcribed or made into RNA. After that, when this terminator region comes, the polymerase will know no more transcription. It will stop there immediately. So and then where this terminator sequence is also transcribed, and also this little part is also transcribed. So this is where the gene ends, and apart from the gene there is some more, a little bit of a sequence, and that this is known as the transcription termination site. So a particular sequence comes or particular factor comes where it understands, okay, this is the place where I have to stop transcription. So transcription stops right over here. This is how it goes. And so this region is known as, this part, this light green part is the actual gene which is going to be made into a protein. This is the gene which possibly is going to be made into the protein. This is in bacteria. We are talking about bacteria now. In eukaryotes it's a bit different. So this is going to be So this is known as the downstream of the gene region. Now, before we come to this region, now let us go let us go come to this region after seeing the photo. So promoter is a sequence upstream of the start of the gene that encodes the RNA. The RNA polymerase interacts with the promoter. The way the RNA polymerase interacts defines the direction of transcription and thus dictates to the enzyme which DNA strand is the template strand and where transcription is to begin. Then you have the RNA coding sequence itself, that the particular gene is located, and a terminator which specifies where transcription stops. So after comparing several let us have a look at a bacterial promoter. Let's see if I can find a picture of a bacterial promoter. So have a look at the bacterial promoter region. This is the let's take this blue color. So this is the promoter region. This much region, it is the promoter region, and in the promoter region, so this is the place where you had seen, this is the +1 site. This region is transcribed into the messenger RNA. This region is transcribed into the messenger RNA. So this is the -1 region. There is no 0. So at the -10 region is the place known as the -10 box. That is also known as the Tata box or the Tata consensus at the -10 region. Then around 17 ±1 base pair ahead, you have the -35 box. Some people also call it the many people call it the GC box, or the best to speak of it as the -35 box, and it has got a TTGACA consensus. So this is the essentially this is the promoter region of a bacteria or a prokaryote. So this is how it is the case in prokaryotes. So let us get back to our place. So they here there are so in E. coli you can find there have been critical for initiation of transcription these regions. The first one, as I had shown, is the -35 region, and the other is the -10 region. It is the -10 region, and that is 35 and 10 base pairs upstream from the +1 base pair. So they are known as consensus sequence. In the -35 the sequence is TTGACA, and in the 10 it is TATA, T-A-T-A-A-T. You have to remember this. This is also known as the Pribno box. Some people call it the Tata box. In bacteria, there is only one type of RNA polymerase is found, and it is made up of, it has got a core enzyme, and it is and the complete is known as the holoenzyme. So holoenzyme is made up of the core enzyme, and that has got 2 alpha units, 1 beta unit, and 1 beta dash polypeptide. So the core consists of 2 alpha, 1 beta, 1 beta dash, and that's it. That's the core enzyme. Along with the core enzyme, there is another polypeptide which is there. It is known as the sigma factor. The function of the sigma factor is that the sigma factor ensures that the RNA polymerase binds in a stable way only at promoters. If there is no sigma factor, it can't recognize the promoter and bind to the promoter sequence. So it is therefore very important that a sigma factor is present. So the molecular weight this is the eraser. Let me erase it. ठीक है, yeah. So the molecular weight of 70,000 kilodaltons or 70 kDa, it is the molecular weight of the sigma factor. The promoter of most genes in E. coli have the -35 and -10 recognition sequence. So these promoters are recognized by a sigma factor with a molecular weight of 70,000 kilodaltons. So let us read only this part itself. So now let us go to the next figure 4a. This is figure 4a. Let's see if I can increase the size. If it is let's keep it smaller only, that's better. So the title of this figure is Action of E. coli RNA polymerase in the initiation and elongation stages of transcription. So in initiation, the RNA polymerase holoenzyme is the complete enzyme which has got 2 alpha units, 1 beta unit, and 1 beta dash unit, as well as the sigma factor. So that is the holoenzyme. Holo means complete. The core enzyme does not have the sigma factor. Remove the core enzyme. With this dark yellow one, it is the sigma factor, and light yellow it is the core enzyme. Okay, so what happens now is, you can see the 5' to 3' strand is always like that. It is unwound. It is in a wound state, and the RNA polymerase, it recognizes that this is the promoter region which has got the 2 consensus, -10 region, the Pribno box, and the -35 region. And on the basis of that, the sigma factor recognizes that this is the promoter region and comes and binds to this promoter, which is colored in green. So now the DNA is not yet unwound. Recognition takes place. So the RNA polymerase holoenzyme first recognizes the promoter at the -35 region and binds to the full promoter. So everything is closed. The DNA is wound and all are bound to the promoter. This is known as the closed promoter complex. This, if they ask the question, what is a closed promoter complex? This is the closed promoter complex. The sigma factor is bound over there. So you can see that they are showing that this region from here to here, this is the -35 region, and this region is the -10 region. So let's go to the next picture. As initiation continues, RNA polymerase binds more tightly to the promoter at the -10 region. At the -10 region, accompanied by a local untwisting of the DNA at that region. So, as you can see that this region is unwound over here. This region you can find that is unwound. So, this region is unwound. That is done by this RNA polymerase. At this point, the RNA polymerase is currently oriented to begin transcription at +1. So, when it is unwound, the -35 region is here, and this is the -10 region. So it is unwound, and now it is in a perfect place to start transcription. The initiating nucleotide is the +1 nucleotide. So the first nucleotide triphosphate comes and sits over here. This complex is known as the open promoter complex. So you can expect questions coming. What is closed promoter complex? What is open promoter complex? So after 8 to 9 nucleotides have been polymerized, the sigma factor dissociates from the core enzyme. So after an RNA, this is something like 8 to 9 nucleotides, an RNA DNA hybrid which is formed has been synthesized. This sigma factor gets released. This is the next step. Direction of transcription is in this direction. Okay. As next step is, as the RNA polymerase elongates the new RNA chain, the enzyme untwists the DNA ahead of it, keeping a single stranded transcription bubble spanning about 25 base pairs. About 9 bases of the new RNA are bound to the single stranded bubble, while the remaining existing the enzyme in a single stranded form. So around 9, this is the bubble. This whole circle is the bubble. This is the bubble. This is the bubble. So in this bubble, what they are suggesting is that around 9 bases are here in the bubble. In this, around 9 bases in the bubble, and this is all coming out. And around approximately about 25 ahead, around 25 are kept unwound. এগিয়ে যাচ্ছে 25টা করে অ্যাটলিস্ট যাতে খোলা থাকে, আর 9টা এই বাবলের মধ্যে থাকে RNA DNA hybrid. So this is how the RNA elongation, mRNA elongation takes place. So this, now it has gone from the promoter to the pink region, which is the RNA coding sequence, and this is the mRNA which is being made here. So this is how transcription initiation takes place and elongation takes place. Now, how does transcription termination takes place? There are two ways by which it takes place. First, termination of an RNA change is signaled by terminator sequence in the DNA. That is, in E. coli there is a protein known as rho protein. This is how a rho protein is symbol of rho. Plays a role in the termination of transcription of some genes. So there are different types of terminators. One is the rho dependent terminator. They are also known as type 2 terminators, and of course you have the rho independent, without the help of rho. They are known as type 1 terminators. So what is They consist of an inverted repeat sequence about 16 to 20 base pairs upstream of the transcription termination point, followed by a string of 4 to 8 A to base pairs, A to T base pairs. So this is how this is on the DNA, template DNA. This is the template DNA. This is what happens. You have an inverted repeat. So what is an inverted repeat? This region. This is complementary to this pink region here. This is complementary to this pink region. So when this is transcribed into the mRNA, you have this region, yellow region, is complementary to this region. And if they are complementary, what will they do? They base pair. And you have a hairpin-like structure. So this is how the structure takes place. So the polymerase transcribes the termination sequence, which is part of the initial RNA coding region. So this is in the RNA region which is taking place. Because of the inverted repeat arrangement, the RNA folds into a hairpin loop structure. So this is the hairpin loop structure, termination hairpin loop structure in the messenger RNA. Now what happens? The hairpin causes the RNA polymerase to slow and then pause in the catalysts of RNA synthesis. So then it stops. The string of EU nucleotides downstream of the hairpin destabilizes the thing, and the RNA polymerase dissociates from the template. It leaves the template and it leaves the DNA, and the transcription ends. So now the row dependent, we were talking right now of the row independent terminator, which has got a hairpin. This row independent terminators have a hairpin. Now let us go to the row dependent terminators. They are C rich, they have lots of C's and less G's, and they have no hairpin structures like those of row independent terminators. Termination is as follows. Row binds to the C-rich terminator sequence in the transcript, and transcription and RNA polymerase is dissociated. Basically, that's what happens. Row comes and binds to the row dependent terminator, and end of transcription. The RNA polymerase leaves. And in row independent, in the mRNA, a hairpin is formed and the RNA polymerase leaves. So there are two types of transcription termination. So this basically it ends the chapter.